Hello biology, and welcome to a quick video going over how your cells regulate blood sugar. To do this, I'm going to be talking a few ideas and then going to a video where I have drawn how two different cells undergo a negative feedback loop to help regulate our blood sugar levels. Generally, you should know that our cells in our body will use insulin to help absorb and store glucose after a meal to store it primarily as glycogen. And that's going to be reducing our level of glucose in the blood. Now, in order to stimulate this, we need to first produce insulin, which is going to be coming from our pancreatic cells in our body. Our pancreas is an organ somewhere in the mid-thoracic cavity, and your beta cells in that pancreas are going to be making insulin as a result of high blood glucose levels. So this shows a positive-negative feedback loop because when we make insulin, we're going to store blood sugar, and we use blood sugar as a stimulus to make insulin. And we can see this on a daily basis when we look at insulin levels, which are in blue, spiking alongside red, which is blood sugar levels. So when you eat a meal, your blood sugar will go up and insulin will go up as well. I'm going to be posting this video and also the picture going alongside it to help you with these two drawings. Note that you're going to be doing one page on the left side and one drawing on the right side. So here we go. Apparently this captures sound, so at some point I'm going to be interrupted. That's okay. So one picture on the left is going to be your pancreatic beta cells, and the one on your right could just be a muscle cell. The red stream in the middle is going to be the bloodstream. You could draw that on one or both of your pictures. Your pancreas also has alpha cells, and alpha cells make your body also has alpha cells in your pancreas, and those alpha cells make glucagon, which helps regulate your body when it doesn't have enough blood sugar. So what I'm drawing right now is our first part of pancreatic cells, which is going to be a protein in the membrane. That's why I'm right now drawing the alpha helices. And this is going to be a channel protein or a transport protein. It's a GLUT protein, which stands for glucose transport, GLUT. I like to think if you eat a lot of food, you're a bit gluttonous, which means you need a lot of GLUT. And remember, carbohydrates such as glucose, C6, H12, O6 are solid, polar, and large, so they need something like a protein to get in and out of the cell because they can't simply just get between the phospholipids. Now, when you eat a lot of food, say a bunch of M&Ms, you have a high concentration of blood sugar. So by default, by diffusion, you're going to want that sugar to enter your cells. So they're always going to naturally flow in through these GLUT transport proteins. Once we get that glucose into our bodies, as we'll be talking about in our third unit this year, you have enzymes, which are proteins that are going to bind to that glucose and undergo a couple chemical reactions called cellular respiration. We're going to be breaking apart that glucose almost like wood stoves and logs, and the general product of cellular respiration is making ATP, which is a small, tiny chemical molecule which contains phosphates, which I'm drawing right now up above. Phosphates are always a phosphorus with 1, 2, 3 single oxygens and a double oxygen, meaning it is incredibly negative. Remember, those negative molecules are going to be interacting with other things. So we get glucose into the cell, do cellular respiration, and then we're going to use those phosphates to do some work. And the work that we're going to be doing is we're going to be binding those phosphoruses to another transport protein. This is a potassium ion channel, which means it's a protein that looks like a tunnel, and it allows positively charged potassiums in and out of the cell. That probably means that the inner lining of that tunnel will have a lot of R groups that are negatively charged, because potassium is a positively charged atom. When we have a lot of sugar in our body, some of those phosphoruses or phosphates made from cellular respiration are going to be bound to the potassium channel, causing it to close. And the reason why is those negatively charged oxygens will interact with negatively charged amino acids, which cause the whole protein structure to change ever so slightly. Basically, we're closing the door by just swinging a few latches shut. What this means is the potassium that normally could flow out of our body is stuck. It can't get out because they're charged solid molecules. They can't interact with the fats. So we're going to be accumulating a bunch of potassium in the cells, and those potassium molecules are going to do something to another protein. This protein that I'm drawing right down here is called the calcium voltage gate. That's normally shut off. Normally, calcium, which exists outside of our body, can't flow in through the cell membrane, again, because they're solid and charged, so they can't enter. Normally, this protein is closed. However, if you bind a potassium ion, a positively charged potassium, to that protein gate, that positive charge is going to cause the entire protein to change its shape as a result of positive and negative interactions, opening the protein. So now calcium can flow in through the open tunnel, allowing us to have calcium within the cell. That calcium is another positively charged molecule. Notice I accidentally erased my cell membrane, so I'm going to redraw that right now. Mistakes happen. But these calcium ions are also positively charged, and positive and negative interactions between proteins and other molecules allow us to change shape, and that means we can turn things on or off or make them do work. Within our pancreatic cells are a bunch of insulin proteins already made. That's what the I stands for. It's a polypeptide protein with a very specific structure and shape. Calcium binds to those transport vesicles. That's what those black circles represent. And the calcium spurs the insulin to be dumped out of the pancreatic cells and into our bloodstream. So we're releasing a hormone as a result of that high glucose concentration flowing in. So what I'm drawing right now in the bloodstream is a bunch of I's and a bunch of sugar molecules. You just ate a Snickers bar, you have a lot of sugar in your blood, and as a result, your pancreas is starting to release insulin. It's time to do stuff with that blood sugar. High levels of blood glucose in your body can be very dangerous to a variety of cells, especially if they can't get the blood sugar in, which is why insulin is so important. So what I'm drawing right now in the second cell, the muscle cell, is an insulin receptor protein. It's another protein embedded in the membrane of muscle cells and has a very specific structure that will allow insulin and insulin only to bind to it at that active site. I know I'm in the way right now, so I'm going to show that in just a minute. Hopefully I showed this in a minute. That would be very awkward if I did it. All right, so let's pause here for just a second. If you notice, you have insulin binding at the exposed site of the protein. These green lines are hydrogen bonds between positive and negatively charged regions of both the receptor protein and the insulin. Sort of like a lock and key, they're going together, which is going to cause change in shape to the insulin receptor. The insulin receptor is now going to activate proteins or molecules inside of the cell as a result of that change. Sort of like all the things that you can now do inside of your house once you unlock the door. IRS is that first secondary messenger, and IRS is going to go activate another protein, which is why that arrow was there. And that green protein that I'm about to draw right now is known as P3K, protein 3 kinase. It's another enzyme that's going to be used to activate other molecules. We're also turning this P3 kinase on by using a phosphate. And that phosphate is going to bind to the protein structure, causing it to change its shape because of that negative interaction. Remember, positive and negatives are interactions between proteins and other molecules that can cause them to change their shape. P3K is going to go act on the final secondary messenger, which is another protein. It's also going to be activated using a phosphate. And as soon as P3K is activating AKT, it goes dormant, so we have a negative positive feedback loop. P3K can only influence AKT, and once it does that, it's turned off. AKT is going to go and do two different cellular responses as a result. The first is it's going to go find transport vesicles that have already been made that store a transport molecule ready to be released. And this is GLUT4. GLUT4 is another type of glucose transport that's only released and made into cell membranes when blood sugar levels are high. And we make a lot of it. AKT is going to go activate those transport vesicles to put GLUT4 in the protein membrane. What that means is, I think I'm writing the word transduction right now, which is the second step of cell signaling. This is our response. What this means is glucose can now flow into the cells, reducing our blood sugar levels. Now, the second pathway that AKT operates on is it activates an enzyme known as glycogen synthase. Where enzymes are proteins that have a really specific structure, and that mouth is what's called the active site that allows it to bind to a very specific thing. Glycogen synthase is an enzyme that can only interact with glucose, and what it does is it binds specific molecules of glucose together to form a polymer of sugars called glycogen. Glycogen is a long chain of glucoses stored together in your muscle cells that allow you to take lots of sugar from a pasta party or too many pieces of candy and store it for later. So here is our result. Step 1, you eat a bunch of sugar, and some of it makes its way to the pancreas and the bloodstream and goes through GLUT transport proteins. Step 2, that glucose is burned to make ATP in cellular respiration. The phosphates in ATP are very negatively charged. Those phosphates help close the potassium channel, which prevent potassium from leaving, and they too go and interact with the voltage gate. Step 5, that potassium or that calcium activates insulin to be released into the bloodstream, which then go and binds to receptor proteins in different muscle cells. That causes AKT to be activated. AKT tells GLUT4 to get embedded in the cell membrane, allowing us to absorb glucose in all of our cells. And then glycogen is made as a result, lowering our blood sugar levels. I'm sorry that last bit was a little bit blurred, so we'll just stop here for a second. 1, 2, 3, 4, 5. Reception, transduction, response. Reception, transduction, response. Now, because of this, what's going to happen directly in step 9 is we're going to be taking a bunch of glucose out of our cells or out of our bloodstream. Because we have no glucose, no more will be entering pancreatic cells, meaning that this step and this step and this step and this step will no longer be happening, so insulin won't be being secreted and signaling muscle cells to absorb glucose. This is why we go up and then we go down with our blood sugars. The insulin is released as blood sugar levels rise, and then we control the blood sugar, showing a negative positive feedback loop. And there's not a negative positive feedback loop. This is a negative feedback loop, but something positively occurs which makes us negatively control it in a way that we go back to homeostasis. I hope this video has been helpful. I'm going to post this image in Schoology. Let me know if you have any questions.